Alright, so this is chapter 13. These are the problems. I think it's better to sort of lecture by reading problems and solving them. And then that sort of puts you in the frame of mind of sort of solving problems. And when you're doing these on your own, you should look at the problem and not, I would start the whole chapter with looking at these problems. And then when you need to figure out the answer, just kind of go look it up as you go. So this chapter is all about transcription, which is DNA being copied into RNA, and that's part of the overall flow of genetic information. And this is transcription here, and we often abbreviate that TXN, so just to sort of make it a little bit quicker. So we're talking about transcription, DNA going to RNA, and so we need to think about the structure of RNA. So this first question is just what's an RNA nucleotide, and compare it to a DNA nucleotide. So let's start with DNA, since that's the one we know already. And we know DNA has a sugar, and in this case the sugar is deoxyribose. And that's because there's no oxygen at that position right there. So what position is that? That's the 1', 2', 3', 4', and then we get another carbon here, 5'. These little primes don't want to stay on there. And so the 2', so sometimes they even call it 2' deoxyribose. And so that's the sugar, and then we can have 3 phosphates off of the 5' carbon, and then we'll have a base off the 1' carbon. In DNA, that base is either A, G, C, or T. And then ribose, a ribonucleotide, is A, G, C, or U. It has an oxygen here, so it's a ribose sugar, not a deoxyribose sugar. And everything else is pretty much the same. Got a 3' OH, so you always have to have a 3' OH, that's important. So in all of these things, DNA, RNA, you need to have a 5', you have to have 5' phosphates, and you need to have a 3' OH. And then in RNA you have a 2' OH, and in DNA you just have an H at the 2' carbon. So those are the major differences. The other thing, when you put these together, DNA, that's one of my worst helixes. DNA is a double helix, and RNA is single-stranded. So DNA is double-stranded, RNA is single-stranded. Come on, pen. And that allows RNA to fold up into different kinds of structures. And so that's important. And we'll look at that a little bit more later. So that's the different structures of the two types of nucleotides. And so this is an RNA nucleotide. And so it's a nucleoside triphosphate, really, because it has 3 phosphates, where this is a D-NTP. This is just an NTP. You could call it an RNTP if you wanted to just remind yourself which one was which, but usually it's just NTP. So what kinds of RNA are there? There are 3 major types of RNA, and it's actually probably useful to think of, so here, if this is DNA, all of these RNAs still come from genes, and so we'll, just to generally draw those like that, and we can make from that, we can make a messenger RNA, a different gene somewhere else might make a ribosomal RNA, and a different gene somewhere else might make a tRNA. And so a messenger RNA eventually is a long single-stranded RNA, and if it's a messenger RNA, it has an open reading frame in it, So this information that codes for the protein, and this bit of information that codes for protein is called an ORF, and ORF stands for Open Reading Frame. And so that's the segment of the RNA that codes for protein. All right. So that makes this because it codes for protein, makes it a protein coding gene. So protein coding genes make messenger RNAs, and then they eventually code for a protein. And this protein will have some three-dimensional structure and look precisely like that. Ribosomal RNA, this is ribosomal. And this is transfer RNA. These do not code for proteins, so these are non-coding RNAs. And it turns out there's other types of non-coding RNAs, but this is asking for the major ones. And so I'm pretty sure they're just going for these three major ones. And so these guys are the ones, so ribosomal RNA, you should Google ribosomal RNA and look at the actual structure. And that's not exactly what it looks like, but the idea is that it's got a complex folded structure. Transfer RNA has what they call a cloverleaf structure. And it has an anticodon. And that anticodon actually matches up with the messenger RNA during translation. And so the function, so it's not asking for the functions quite yet, but the ribosomal RNA is part of the ribosome. The transfer RNA sort of carries an amino acid would be connected to that transfer RNA, and then during translation, the transfer RNA matches up to the mRNA and brings the appropriate amino acid to the ribosome. So those are the three major classes. And the difference is mRNA codes for protein. Ribosomal RNA and tRNA are both involved in translation, but they themselves don't code for proteins. And when we're talking about transcription, we talk about a transcription unit, and that is just the region of DNA that gets transcribed into RNA. And they look a little bit different in bacteria and eukaryotes. In bacteria, there are some similar things, and then down here, so you have a transcription start site. So it's imaginatively named. And we call that a TSS. We've got a terminator. And then we have the coding region. So whenever you see the word coding, we're talking about coding for protein. And then upstream of the transcription start site will be a promoter. And that, I guess we could consider that part of the transcription unit. This overall thing is a transcription unit. You're going to make an RNA out of that transcription unit. And just to stay in the habit, we always draw RNAs 5' to 3', just because that's the direction that they're made. So that's pretty much it. Another thing I'll add is that in bacterial genes, so transcription unit, it's kind of another word for gene. We talked about genetically identifying genes before, and this is like thinking about a gene at the molecular level. So you can think of the gene at the molecular level as the part of DNA that gets transcribed into RNA. And this is a protein coding gene that we're talking about here. Although you could have a transcription unit, like a region of DNA that gets transcribed into tRNA would also be a transcription unit. it would also have a promoter and everything. But in this case, if we're going to make protein in bacteria, genes are often organized so that you make several proteins from the same RNA. So you have 3 different open reading frames. All right, and so that's a little bit different than what we see in eukaryotes. I'll draw in a minute. We call these RNAs that have multiple open reading frames polycistronic RNAs. And that's just because another old-fashioned word for gene or for protein coding region of a gene, they used to call them cistrons. And so this is called a polycistronic RNA. But the main parts are the promoter, the coding region, and the terminator, and the transcription start site. Alright, so we got a little typo there. Sometimes I get a little carried away with apostrophes. Just when drawing things quick. So a transcription unit in eukaryotes also, and we'll make this blue. So how do they get way down there? So in eukaryotes, so it's always key, a lot of times in questions, things get distinguished between prokaryotes and eukaryotes, and so it's important to pay attention to those distinctions in questions. If you read them quickly, you say, I just drew a transcription unit. Why? This isn't the same. So we have a transcription start site, and it's always given a +1, so that's TSS, and there'll be some kind of terminator down here. But the termination is a little bit different in eukaryotes, so we'll go ahead and draw this sequence out here. My pen doesn't like to make those little A's. We'll also have a promoter. I just draw these in red just to, I don't know, because these are a little bit different. We got a promoter, and then we may have an enhancer. There might be some other sequences that are to the left of the transcription start site that are important. We will draw in this little symbol. That just means that this is far away, or at least it can be far away. So that's what that means, that there's a lot of extra space in between those two things. So we got a transcription start site, a promoter, enhancer. This thing is called the poly A addition signal. And we'll get into that a decent amount in chapter 14. And then we can also throw some boxes in here, but these boxes are not, they don't code for their own proteins. In bacteria, those 3 boxes, and it could be more than 3 boxes, but those boxes code for proteins. They're their own open reading frame. In eukaryotes, we're going to transcribe it a little bit further. We're going to transcribe the gene into RNA, but now these boxes are exons. And the stuff in between are introns. And then we get RNA processing and splice the exons together. We still have this sequence in the RNA. And then when we do processing, and we'll get into this more in chapter 14, but we get a poly A tail. So this is just the signal that says, okay, add a poly A tail now, and add it down that way a little bit. And then we'll also add some kind of 5' cap on the end. So this is always the 5' end and the 3' end. Alright, the only other terms I'll throw out here is that this is always called the upstream direction. So anything to the left, we almost always draw it left to right, but it's really to the left, or it's really to the 5' end of the gene, and the 5', and we relate it to the 5' end of the RNA that is made, and then down in this direction is downstream. So you can think of transcription flowing in this direction, and it's flowing from upstream to downstream. But the main parts, as far as the question goes, are enhancer, promoter, transcription start site, exons and introns, and our termination signal in eukaryotes is this poly-A addition signal. Substrate for RNA synthesis is a Alright, so what is the substrate for RNA synthesis? It's going to be an RNA nucleotide. So RNA synthesis means we're going to take individual RNA nucleotides, and an RNA nucleotide looks like this: 2' OH, 3' OH, and 3 phosphates. Keep in mind these phosphates have oxygens involved, but we abbreviate them like that. And then we're going to so we got another RNA nucleotide up here. And this is just sort of showing the basic reaction of linking two RNA nucleotides together. And this could be some other nucleotide. And it's probably got its 3 phosphates here. And so we're going to take this phosphate and we're going to add this to the 3' OH of this nucleotide. And so when we do that, what we end up with is 2 nucleotides linked together. And so this OH is here, and it connects to this phosphate. And so this one would be the U nucleotide, because it's being added there, and this would be the A nucleotide. So just like in DNA synthesis, we need to add the 5' phosphate of the RNA nucleotide to the 3' OH. So this is the 3' OH. We lose these phosphates off as inorganic phosphate. And that's how it goes. How is it modified? I guess the only way it's modified is it loses these phosphates. And this guy would still have 3 phosphates on it. And then you'd have your 3' OH here, 2' OH here. The 2' OH doesn't really play a role in synthesis, it's just kind of there. But the 3' OH is there. And so what we'll see is we'll have a DNA template. And then we're going to add nucleotides. And we're going to link those together by phosphodiester bonds. And we're going to add those in the 5' to 3' direction, because we have to add it in this way. We have to take this 5' phosphate and add it to that 3' OH. Just like DNA replication. It's exactly the same. We'll see in a little bit. There's a little bit of difference. The main difference is that you don't need a primer, but we'll come back to that. But it's 99.9% the same as DNA replication, or DNA synthesis, at least that part. Holoenzyme of bacterial RNA polymerase. Holoenzyme just means the complete enzyme. And so the holoenzyme of DNA polymerase Alright, so the holoenzyme of bacterial RNA polymerase. One of the key things here is that in bacteria, this is mainly E. coli, there's just one RNA polymerase. And it's just called E. coli RNA polymerase. So RNA polymerase is the enzyme that makes an RNA polymer. So that's what it does. And it has 5 subunits. And exactly how they're arranged doesn't really matter. 2 of them are alpha. Well, I mean, it does matter, but for this drawing, it doesn't matter. And then there's beta, and then beta prime, so that's similar to beta. but it's not exactly the same. And then this one's omega. And so this is the core enzyme. And so this core enzyme with 5 subunits, so this is the core, it gets joined up with another one called sigma. And so this is the core plus sigma is the complete enzyme, and that's called the holo enzyme. And so there can be different sigmas in the cell, so you can have some variation in the types of RNA polymerases that there are. So there's one. I mean, they're all basically the same, but it might have a slightly different sigma subunit, and that subunit is just each of these subunits is a different protein, and that sigma subunit might target the RNA polymerase to different genes. So some genes might get transcribed by this RNA polymerase, and other genes might get transcribed by that RNA polymerase. Now in eukaryotes, we're not going to worry too much about the structure, but the main difference here is that there are 3 different RNA polymerases in eukaryotes. And so this is just pretty sort of memorizing the basics here. So we've got 3 major ones. And each of these has a bunch of different subunits to it. And this is the main one that we talk about. So it makes mRNA. It is the main RNA polymerase, and it transcribes protein coding genes and makes mRNA. RNA power 1. So, and then the other major types of RNA are tRNA and ribosomal RNA. So you just have to remember that this is the main one, RNA polymerase 2. That's the only one we really talk about that transcribes protein coding genes into mRNA. And then we've got 1 and 3, and 3 does tRNA. So that's pretty easy. So 3 T. So we can remember that. And this does ribosomal RNA. Now there's a bunch of other RNAs in the cell, small nuclear RNAs, siRNAs, and stuff like that, and various ones of those. Kind of depends on the RNA that gets transcribed, which RNA polymerase works. But just to keep it simple for this question, those are the 3 RNA polymerases, the 3 different RNAs they code for, or they transcribe, and then in bacteria there's only one. So ribosomal RNA genes, tRNA genes, mRNA genes all get transcribed by the same RNA polymerase in bacteria. Not sure how this happened. Somebody's been scribbling on my question. I also don't know why it keeps jumping all the way to the end, but let's get rid of all these marks. Alright, I'll quit doing that. Technology, technology, technology. What are the 3 basic stages of transcription? Well, there's acceptance, denial, accept. No, wait a minute, that's something different. Hold on a second. The 3 basic stages of transcription. This kind of cracks me up sometimes. All these different processes. So we got 3 basic stages. We got the transcription start site. So that's your TSS. Here, I'll write it up here. And then stage 2, then stage 3. So we're going to have the beginning, and we're going to call that initiation. Then we have elongation and termination. So termination. Initiation, elongation, termination. Every single process that exists has a beginning, a middle, and an end. And you can call beginning initiation, you can call the middle elongation, and you can call the end termination. So what happens at each stage? Well, at stage 1, RNA polymerase binds and begins to transcribe. All right. Elongation, RNA polymerase is kind of gone further down and is transcribed a little bit. And then termination terminates. And the RNA polymerase falls off and releases the RNA molecule. So that's kind of the basic things that happen. It's always important to think about the direction of transcription. And we're also, this DNA is double-stranded, and so as we look at this in more detail, we'll look at what happens to the DNA and what strand gets copied and all that kind of stuff. Alright, so that's just the basic answer to that question. Now draw a typical bacterial promoter and identify any common consensus sequences. So in bacteria, the promoters have similar sequences. And it's important now, just that what I really want to get across when we're talking about transcription and translation is that this is a flow of information, and that DNA contains information. That's mainly what it does. It doesn't really have any function, it doesn't really do anything, but it stores information, and then the cell with its different enzymes like RNA polymerase and all these different enzymes can look at that information and extract that information. And so in bacteria, the information that tells the cell that this is a gene and it's time to transcribe it, that information is contained in the promoter. And there are two important regions of the promoter, one at about -10 and one at about -35. So minus means relative to the beginning. So if +1 is the beginning, I don't know why they don't call that 0, but they call that +1. That's the first nucleotide. So if we transcribe, the very first nucleotide counts as the first nucleotide, so +1. I guess I don't even know if there is a 0. I think they might even skip 0 and have a -1. But the general idea is that at position -10 and at position -35, relative to the beginning of transcription, there's something called a consensus sequence. So a consensus sequence just means they've lined up a whole bunch of different genes, and they look at these promoters and they compare the sequence upstream of the transcription start site. So if this is +1, they just compare all these different things using a computer and say, is there anything in common? And there's something that's similar in all the genes in this area, and there's something similar in this area. Whereas that area it just varies, it's just random. But this area is conserved, meaning that the sequence is the same. And so it's just sort of a rule in when you're comparing different DNA sequences from different genes or from different species. If the DNA sequence doesn't change, or if it doesn't change much, then it's said to be conserved, and that means it has some kind of a function. So one way you can identify that function is by looking at by comparing a bunch of different sequences and coming up with what's called a consensus sequence. Now, I certainly don't want you to memorize this necessarily, but the sequence at -10 is TAT. And just to give away all my secrets, I'm looking at it right now on the screen, and I'm not memorizing it. The only one will have you memorize one sequence maybe in eukaryotes. But that's what we call a consensus sequence. And so there's a question later on that shows you a little bit more about consensus sequences, but that's like what that means is that's the most common sequence that's conserved in promoters of bacterial genes. So the same thing with terminators. Terminators have some kinds of sequences that are conserved. So the different types of terminators are get a little room here. So we're just sort of looking at the structure of bacterial genes. Transcription start site, promoter. Down here there's a terminator. And there's a couple different kinds. There's row dependent, and there's row independent. And it's just different genes have different versions of these things. This is the different types of terminators. Row is a protein. So that's a protein that binds the RNA and causes transcription termination. And then there are these row independent ones that don't require a protein. And typically so the row independent ones, instead of having a protein that binds them, they form what's called a hairpin structure. And so these row independent ones, after transcription, so we'd have DNA, we'd have RNA polymerase, and then we'd have our RNA that's coming off of this. But actually there's a sequence in the RNA that forms some kind of a hairpin. And this hairpin terminates transcription. But it doesn't require a protein. The hairpin itself somehow causes transcription to start. So in row independent, there's secondary RNA structure. So we looked at RNA structure and we said it can fold up into different shapes, and that's a hairpin, and that hairpin causes the RNA polymerase to fall off. The row independent or the row dependent doesn't have a particular sequence. I mean, it has a particular sequence, but it doesn't form secondary structure. It just forms a site for a row protein to bind, and that row protein causes transcription to stop. So that's basically it. Row dependent, row independent. We may come back to this. So one thing we'll say about this is this guy here is called an attenuator. And it's one of the first examples of secondary structure of RNA actually doing something. And so now, question 11. So this is asking about transcription factors. And so transcription factors are factors that affect transcription. And so we're going to go into this in a little more detail than the book. The book's answers to these things are not always the greatest in detail. So bacterial gene, this is just sort of the general thinking, is we have a promoter, and we have RNA polymerase, and the RNA polymerase can basically bind the promoter and transcribe. That's its job. And it'll probably have a little sigma subunit on it, and sigma helps the RNA polymerase bind the promoter and transcribe the gene. So it's pretty simple. There's not a whole lot of different protein factors that are involved. When they start talking about general transcription factors and transcriptional activators, in bacteria, I guess we'll just keep it like this. In bacteria, there's not really any a lot of transcription factors, but you might have an activator protein. That was supposed to be a different color. You might have it. Come on, be a different color. You might have an activator protein that helps RNA polymerase bind. Now, in eukaryotes, it's much more complicated. And a lot of times, I mean, that's kind of the, I mean, that's the general rule. Eukaryotic cells, genes are more complicated, cells are more complicated. We've got a lot more, tissues and organs and stuff like that, so we're way more complicated than bacteria. So eukaryotes, not eukaryotics, eukaryotes. When we have a gene, we're going to have our transcription start site here, and there will be a promoter. But there's a bunch of transcriptional activator proteins. Well, first of all, these are general transcription factors that have to bind at the promoter. And these things are called TF2, TF stands for transcription factor. And there's a lot of different ones. A, B, there's no C for some reason, D, E, there's a whole bunch of these. And those things have to bind at the promoter, and then RNA polymerase can bind. So these are general transcription factors. And these bind at the promoter, and they pretty much bind at every promoter. So every promoter needs to have these general transcription factors bind to them. And then in eukaryotes, down here we have other DNA sequences that control transcription, and these might be enhancers. And these enhancers will cause genes to be expressed only at certain times. So you might have a specific kidney enhancer or a liver enhancer or a brain enhancer. And then there are activator proteins that bind these enhancers. And just to give you a little preview of where we're going with this eventually, I should probably try to keep my colors somewhat similar, is these activators, they help, the DNA kind of loops out, and these activators, they help these transcription factors bind to the promoter. So these guys help that happen. So activators activate transcription by causing these general transcription factors to bind, and then RNA polymerase can bind. And in this case, so maybe it's RNA pol2 combined. And these are called TF2 because they're transcription factors that help RNA polymerase to bind. And another term that you'll see for general transcription factors, so these transcription factors are called regulatory because they help regulate gene expression and turn some genes on in certain situations. So that's these guys, sorry. And then these guys, another word for general is basal. So they just mean basic to sort of get the basic. Basal means like the low level, sort of just initial level of transcription. Then regulatory transcription factors kind of crank up that transcription. How are transcription and replication similar, and how are they different? I'm not a big lister, but there are a few important things to keep in mind. There's some certain rules. So similar things, we have a DNA template. The DNA template is always read in the 3' to 5' direction. New molecule made in the molecule made in the 5' to 3' direction. So we're going to have a template. So this is a DNA template. And then we're going to make some new stuff, and we're going to make it in this direction. We're going to make it 5' to 3'. And that's because the new nucleotides have to be linked together by adding the nucleotide onto the 3' OH. But and so that's basically it. You could come up with a lot of other things. They both require enzymes. The enzymes are called polymerases, and so some differences are so we're going to have transcription and replication. So replication involves DNA polymerase. So this is DNA replication. Transcription involves RNA polymerase. Transcription begins at the transcription start site. DNA replication begins at the ORI. Transcription only copies one gene at a time. Now, there might be thousands of different genes that are being transcribed at the same time, but what I mean by this is if we've got DNA, only the gene, I guess a better way to say this is only the genes are transcribed, and so they can go in whatever direction. In DNA replication, the entire so DNA replication is bidirectional. And the whole genome is copied. Only genes are transcribed. And only some genes sometimes are transcribed. So in the brain, only certain genes are transcribed. So let's say here's some other genes. And those green genes are only expressed in the brain, for example, and the red genes might only be expressed in the liver. And so you only transcribe certain things at certain times. In DNA replication, every time the cell divides, you have to replicate everything. So I think that's sort of basically it. I'm kind of looking over at the list on the solutions, and it says it says transcription is subject to numerous regulatory mechanisms. Well, replication can be regulated too, so I don't know if that's the most important thing. The main thing is that everything always, 100% of the time, no exceptions, has to be, you always have to read the 3' to 5' template and do a 5' to 3' synthesis of your new molecule. And know that RNA polymerase does transcription, DNA polymerase does replication. Where they start is important. Transcription starts at the transcription start site. ORI is where replication starts. DNA replication copies the whole genome, and it does it in bidirectional. Now, transcription, I've drawn these arrows in different directions. So transcription can happen in either direction, but it doesn't always happen in both directions. And it will copy a different strand. So if we've got DNA like this, if we're transcribing in this direction, we use the bottom strand as a template. If we're transcribing in this direction, we use the top strand as a template. Oh, the other thing that's important is transcription doesn't need a primer. And so here, in DNA, if you remember, DNA replication needs a primer. And if you might also remember that primase is the type of enzyme it is it's an RNA polymerase, which doesn't need a primer, and so it can start DNA replication. In transcription, the main enzyme is an RNA polymerase, and so it can just start from scratch and just starts right at the transcription start site and starts transcribing. How is transcription different in bacteria and eukaryotes? All the basics are the same. You have to have a 3' to 5' template, make RNA in the 5' to 3' direction, both things need a promoter, that sort of thing. So they're similar in many regards. The main differences are that there are multiple. So now we're talking differences in prokaryotes and just transcription, in eukaryotes. So there's only one RNA polymerase, and there are 3 RNA polymerases. So different types of genes each have their own RNA polymerases. And then eukaryotes are more complex. Requires more regulatory proteins. This just requires RNA polymerase, and just maybe one. Each gene maybe just has one or two regulatory proteins, whereas each gene here might have hundreds of regulatory proteins. And so regulatory proteins are those proteins that help transcription occur. In prokaryotes, we have polycisteronic RNAs. So the overall RNA molecule contains several open reading frames. In eukaryotes, this is the book solutions going into this. This isn't really transcription, but so transcription is just making the RNA and then splicing and adding a poly A tail and a 5' cap. That's unique to eukaryotes, but this is called RNA processing. And so technically it's not really transcription, but if you think of transcription as the overall production of a messenger RNA, there's a lot more steps involved in eukaryotes than prokaryotes. And then the termination system is a little bit different. We've got this poly A addition system in eukaryotes. I guess this just wants me to always do that twice for some reason. Alright, so, and here we have different termination mechanisms, so row independent or row dependent. The main difference that I want you to know here, I would say, is this polycisteronic RNA, and then this is just one open reading frame, and that it's more complicated. That in order to transcribe, you need a whole bunch of initiation factors, or basal transcription factors. You need a bunch of regulatory transcription factors, and these guys have to kind of come together to help transcription start. In prokaryotes, you really just need your RNA polymerase and maybe one or two other proteins that are involved. So these questions are, this first set of questions is mostly descriptive. And then we get into these application questions. And these are the ones where you have to start to think a little bit about taking that information from the other questions and from the PowerPoint and sort of putting it together. And you have to think about these rules, like the template always has to be read in the 3' to 5' direction, and the new molecule is always made in the 5' to 3' direction. So let's take a look at this. So we got some DNA. It's part of an RNA coding sequence of a transcription unit. The bottom strand is the template strand. Well, that's good to know, because it's got 3' to 5', left to right. Give the sequence found on the RNA molecule transcribed from this DNA and label the 5' and 3' ends of the RNA. Now in this case, so if I didn't tell you anything else, and I said what's the RNA product of this transcription of this little chunk of DNA, you would have to know which direction transcription is going. So I could transcribe, not me being RNA polymerase, I could transcribe this left to right or right to left. Kind of depends where the promoter is. So I could have a promoter on the left and then transcribe to the right. I could have a promoter on the right and transcribe to the left. But here it tells you which strand is the template strand. And so whenever you do transcription, you've got DNA. And if I say we're going to transcribe in this direction, that means that one of the strands is the template strand, and it doesn't always get drawn this way, but usually, but it's not a 100% rule. But if we're going left to right, that means we're copying this bottom strand, because if we're going left to right, the thing that we need to copy has to be copied in the 3' to 5' direction. So we call that the template strand. And then this ends up being called a couple different things. I like RNA-like strand the best because it's most descriptive, but you also might see this top strand called the coding strand because it ultimately contains the information that will code for protein, but that can be a little confusing. So if they tell you that this bottom strand is the template strand, that means that the RNA is going to look just like the top strand, because we're going to copy the bottom strand and put in the complementary nucleotide. So I'm going to rewrite this template strand. So there's my template strand. Then when RNA polymerase comes along, it's going to copy this, and it's always going to copy it in the 5' to 3' direction. And then, but when it sees an A in the template, it'll put a U in the new strand. It'll copy that T and give it an A, copy those C's and put in G's, C, G, A, U. Wherever there's an A, it's going to put in a U, G, C, C, A. Alright, so that's this is what the RNA strand would look like. Now you'll notice that the RNA looks just like this top DNA strand. So but except there's U's instead of A's. So up in that top strand where it says ATA, this says AUA. That top strand says GGCG. This says GGCG. Then the top strand says ATG. This says AUG. Top strand says CCA or GCCA, and this says GCCA. So the top strand looks a lot like what the RNA is going to be. Now what I like to do is say, okay, well now what if we transcribe this going from right to left? If we transcribed it going right to left, now we're transcribing it this way. Now the top strand will look like the top strand will be the template, and the bottom strand will be the RNA-like strand. And so I can look at this bottom strand now, that's going to be the RNA-like strand. So now if we're going right to left, we can write in, we're going to put in a U, then GGC, then AUC, so I'm right there, then GCC, then TAT up there, which is going to be UAU here. But now it's really, really important to tell me that that's the 5' end and the 3' end. So now we've copied this, and we've made our RNA. I should have used red. So directionality in transcription, it's not so much left to right, right to left. I mean, it kind of is, but you need to know which is the template strand, which is the RNA-like strand, and that depends on which direction you're going. And then whatever direction you're going, the 3', 5' strand is the template, and the new strand is going to be made RNA 5' to 3'. Now this says for the RNA molecule shown in figure 13.1a, write out the base of the template and non-template strands. So these are good questions to make you think about. What all these terms mean and kind of put them into use. And so the Figure 13.1 shows the actual structure of a particular sequence of RNA, and it shows the 5' end at the top and the 3' end at the bottom. But the important thing is that the sequence is 5' UGAC and then 3'. So it's saying if we make this RNA, what does the DNA look like? It says write out the sequence of the bases on the template and non-template strands of DNA from which this RNA is transcribed. Well, if that's the RNA, then the DNA is going to be the complement of that. So if there's a U there, it has to be an A here, C, T, G, 5'. So that's going to be what our template was. Our non-template, or RNA-like strand, is going to be just like this strand, but have a T instead of a U. So we can kind of work our way back from the RNA to show us what the DNA looks like and what the RNA looks like. Same thing here, and you'll notice I'm picking out these ones in particular. It's all about 5', 3'-ness. The following sequence is found in a single-stranded DNA template. Alright, so sometimes we have a single-stranded DNA template. Assume that RNA polymerase proceeds along this template from left to right. Which end of the DNA template is 5', which is 3'? Well, if it's going left to right, it's template, 3', 5'. Give the sequence and label the 5' and 3' ends of the RNA copied from this template. It's going to be the complement of this. So it's going to be the opposite strand. So where there's an A, we're going to put in a U. TT is going to be AA. GCC is going to be CGG. AGA is TCT. Sometimes it's easier to write it right up there. TCA, AG, and then the opposite of A is U. So where was that? So that was AGU, which was here, here, here. Then T and then 3 C's is going to be A and 3 G's. Then two U's. TAG is going to be AUC. And then AT will be UA. And if this is the RNA sequence, it's going to be 5' going this way. So I'm going to write in, we'll make it green, I'm just going to write in with the complement. Now that I'm looking at this RNA directly, it's easy to say, okay, well that was an ATT. I'm just double checking. GCC, AG, AT, CA, T, CCC, A, T, A, G, A, T. Is that the same as what I had up there? ATT, GCC, AGA, TCA, T, then 3 C's, AA, TAG, AT. So it's the same. This takes practice. I screw it up all the time. If you get a little bit, if you're just kind of cruising along and you kind of switch your mind frame, you can start to mess it up. So just practice as many of those as you can. Oops, I went a little bit too far. Now here's where this idea of a consensus sequence comes from. Gotta get this on here and try to give myself a little room. So this is just going to demonstrate how how you come up with a consensus sequence. This is showing maybe promoters from several different genes lined up, or something like that. So we're just going to compare several different genes and say we've identified this region as being important, and we want to look at these sequences and see if there's anything in common. Now there are some specific rules for exactly how you do this, but we'll just kind of take a look at it, and if there's a majority of one particular sequence, we'll give it that. So just to give you, so in this second position, there's 1, 2, 3, 4, 5. So there's 6 sequences altogether. 4 of them are giving us a G, and so we'll say the second position is a G. But this first position, we have 3 A's and 3 T's, and so at that position, we don't really know. It could be either an A or a T. And then the next position has 4 C's, so we'll give it that. And the answer in the book is wrong. The next one is 4 A's, so that's this one. This one has mostly A's. The next one has mostly T's, and the next one has mostly T's. They just missed one of these, and so that's how you get a consensus sequence. You just sort of look at it and say what's most common at that particular position. So then this kind of gives you information. Let's say this is part of a promoter, and you've got a gene, and you're wondering where the promoter is. You can look for this sequence and say, okay, I see that something that fits this pattern in that area, and so that's probably the important promoter element. Um, 22, this is kind of like comparing DNA replication and transcription. Five properties that they have in common. Okay. They use a DNA template. Depends how you want to parse this out. The templates read 3' to 5'. Let's see. The new thing is made, so template, 3' to 5' template. Molecules are synthesized in the 5' to 3' direction. So the new molecule is synthesized in the 5' to 3' direction. Both use nucleotides as substrates. That's only 4. Let's see. That's kind of those are the main ones. Then differences. DNA pol requires a primer. DNA pol uses dNTPs. RNA polymerase uses just NTPs. DNA pol makes RNA. DNA pol makes DNA, sorry. RNA pol makes RNA. DNA polymerase copies both strands. And RNA polymerase only copies one strand. Alright, so this is pretty straightforward. Draw a diagram of representation of a transcription unit. Alright, and a DNA molecule, and we'll start in bacteria. Draw and then we've got a promoter, terminator, maybe a few structural genes, that code for different enzymes. Kind of the same thing. Eukaryotes. This one was pretty easy. And we got a promoter there. It's pretty much the same thing. So, if that's all they want. Working with molecular blah blah blah. So this is, I just put this in here mainly to emphasize that this is kind of a big area of biology now. So let's say you got this huge stretch of DNA and you want to know where the genes are. Well, we know that genes are the parts of DNA that get transcribed into RNA, and so there are physical methods, actual like biochemistry lab methods, where you can say, well, which part of the DNA is actually copied into RNA. That's pretty commonly done. But you can also, if you have like a new genome, these days you can sequence the whole genome, and it's kind of a big deal to try to do the experiments to figure out where all these genes are. So what you can actually do is design computer programs to look for those sequence signatures that show us the different parts of the gene. And so it's kind of like those consensus sequences. If we know that promoters always have a particular sequence, for example, in eukaryotes, we know that there's a sequence here that's always found in bacterial genes, or I'm sorry, eukaryotic genes. There's something similar in bacterial genes. We could look for that, but it turns out that not all genes have that. That's a pretty short sequence, so it kind of shows up randomly in a lot of different places. We could look for this, but again, that's a pretty short sequence, and it and so this is what we would look for in eukaryotes. We could look for an open reading frame, and so that's kind of what they're getting at here. Is if we have a long stretch of DNA that doesn't code for a protein, the odds of it having this big long open reading frame is pretty low. So if we see a long open reading frame, so I guess we're going to look for a long open reading frame. What that means is look in the DNA sequence for a start codon, and then look for a long stretch of codons that are three-letter nucleotides that you then get to a stop codon. And that's a long open reading frame. And if there's a long open reading frame, it probably has a function. It probably makes a protein that does something. And so that long open reading frame will be conserved. If there's a stretch of DNA where there's no gene, we would expect if there's no reason to conserve a long open reading frame. So you start to see stop codons all over the place. So you might find a start codon and a stop codon, but they might be too close together. So you can look for long open reading frames, and that's really the easiest way to look for genes. Because a protein has to be a certain length, and so you look for genes that are of that length. However, that's complicated in eukaryotes because genes are broken up into introns and exons. And so you might have your open reading frame be in one exon and then end in another exon way down here. And it only becomes an actual open reading frame after you transcribe it and splice it, and then you would have a UG there and UGA here, and then this thing could make a protein. So this is after transcription, splicing, and then translation, we'll get a protein, and we have an open reading frame. But because these introns, they never code for protein themselves, there's no reason, there's no evolutionary pressure to prevent there from being stop codons there. So if you look at introns, you very often find stop codons all over the place. So if you just look at a gene, you can't, I mean, if you just look at DNA and eukaryotes, it's very difficult to just look for an open reading frame. Now, there are ways to look for splice sites and look for, there are computer programs that can say, all right, here's a splice site. If I splice that together, do I now create a long open reading frame? And there are ways to get around that, but it just makes it much more difficult. Because in bacteria there are no introns or exons, it's super easy. You just look for a start codon and you look for a stop codon and you say, is that a gene? Now, sometimes there might be a couple of start codons kind of near each other and you won't know which one of these to pick, and so there are some complications in trying to figure out which is the best one and that sort of thing. But overall, it's easier to pick bacterial genes out of just a random sequence than eukaryotic genes. Through genetic engineering, a geneticist mutates the gene that encodes TBP in cultured human cells. This mutation destroys the ability of TBP to bind the top box, predict the effect of this mutation on cells that possess it. All right, the main reason for this is for you to read this and say, what the hell is DPP, TBP, what's a top box, all that stuff. And also to remind me to point out those types of things. So, typical eukaryotic gene has a transcription start site. So whenever I say draw a gene, start with that. And then it's got a promoter. And in eukaryotes, that promoter, one of the consensus sequences that's found in promoters quite often is this little sequence that is TATAAA, and that's called the top box. And then the protein that binds the top box is called TBP. And that just stands for top box binding protein. So sometimes they just call it top binding protein. And so the TBP is part of a bigger thing. Remember I said there are all these other transcription factors. So really TF2D is this big general or basal transcription factor that binds promoters, and there's a little part in there called TBP that binds the top box. Well, the idea is that most genes, it turns out it's more like 40 to 50% of genes, have a top box and have a top binding protein. So what would happen if you didn't have the top binding protein? You wouldn't be able to bind that promoter, would not be able to transcribe those genes, and the cell would die. Now, if you don't have the top binding protein, all these different genes that you need to transcribe and to make proteins for the cell to live wouldn't be transcribed, and it just wouldn't live. You can't not transcribe all your genes. You have to do that. So probably about 40 to 50% of the genes would not be transcribed. But where do these things keep coming from? All right, elaborate DNA. So maybe that's, I'm doing a little DNA repair here on my document. No idea where all these little things came from. Come on, go away. All right, so as we know, during DNA replication, errors happen, and there are mechanisms for repairing those mistakes. Now, some of those mistakes are repaired during replication by DNA proofreading. If you think back, there's 3' to 5' exonuclease activity that gets then replaced with DNA, etc., etc. And the question here is this the same in transcription? So in DNA replication, the DNA will unwind. We'll make primer going this way, primer going that way, and then if we take a close-up view, sometimes the DNA polymerase makes a mistake, and then it erases that using 3' to 5' exonuclease activity, and then repairs it. Now in transcription, we're going to copy this, and this is the main reason that I do this. Let's say we're making, so how does transcription work? Here's RNA polymerase. It's copying that DNA into RNA. But kind of a key to transcription that I don't think has come up yet in these questions is that we transcribe the gene over and over again. So we're going to make, I don't even know the number, it depends on the gene, but certainly greater than 10,000 RNA molecules. So if there is a mistake here and there, let's say that this made a little mistake, and as long as the mistakes are relatively rare, we wouldn't want to mistake in every RNA molecule, but most of the time the RNA molecule is the same. If there's just some sort of low rate of error, then you're going to make enough functional RNAs for this RNA to go make a normal protein and to make that protein, and you have enough protein for the gene to do what it's supposed to do. However, if you do DNA replication and you make even one mistake, and that mistake stays in the DNA, then every time that gene gets transcribed, it's going to have every RNA will have that mistake in it. And not only that, that DNA will get passed on to the next generation, and so it'll be a permanent mutation in the whole generations following. So there's much more reason or evolutionary pressure to correct mistakes in DNA polymerase than in RNA polymerase. And also, RNA polymerase has to make a whole bunch of RNA in a hurry, and so it doesn't really, it would rather make a few mistakes and just make billions of RNA molecules, and that's just more efficient than just sort of making one RNA molecule and going from there. So that's the basic idea. The basic idea being, one, you make a whole bunch of RNA, so it's okay if you make a mistake in a couple of the molecules, and also the mistakes that you make aren't inherited, and so the damage is sort of minimized at that point. Alright, so I think that covers almost everything that we need to cover in this section, and so we'll go from there. If I can get it to turn off.